Hello everyone, my name is LEE ZHI XUAN. This video is about the free class video for STPM Biology Semester 2 Chapter 9 Transport, special design tool for the STPM candidates who take the biology subjects. So in this video, I will explain about what you will learn in Chapter 9. So in Chapter 9, based on the syllabus, you only learn about two main topics, which are the transport system in humans and also transport system in vascular plants only. You don't need to learn about the transport system in animals. You also don't need to learn about the transport system in non-vascular plants. Only human and plants are in the syllabus. So this is some of the differences between SPM, because in SPM you not only learn about human, you also learn about the fish, insects, right? But in SPM, no, you don't need to learn about insect and fish transport system, you only learn about human. So this is a learning outcome that you can refer from the syllabus.
In this lesson, I'll be using these figures to guide our explanation. If you've already purchased my notes, you'll find the full, detailed explanations written out there to follow along. If you'd like to get your own copy of these notes, feel free to contact me directly.
First of all, we need to learn about the structure of mammalian heart. So this is actually you already learned in SPM. Okay, so this is the basic structure that you need to know you need to know for the structure of the human hearts. So we know that human heart containing 4 chambers, right? So you can see here. Here they're containing 4 main chambers, including left atrium, left ventricle, right atrium, and right ventricle. So between the left atrium and left ventricle, they're containing the bicuspid valve that separate these 2 chambers, and as well as the right atrium and right ventricles. Between these two chambers, they have the separate valve we call as tricuspid valve that separate these 2 chambers. So this is the 4 main chamber that in the heart. So this is that one is the more simple structure. So this is a more complicated structure that you need to know lah. So left atrium is at here, left ventricle, right ventricle, right ventricles, and right atrium. So we know that how the blood flow from the in the heart. So we know that when the lung do the gaseous exchange, they carry the oxygenated blood from the lung into the heart. So when the blood is coming from the lung to the heart, is entering through the pulmonary veins. So the blood will flow from the lung through the pulmonary vein, enter into the left atrium. Right. So in this structure, they're containing left pulmonary vein and right pulmonary vein lah, between these two different position. But the most important thing is that pulmonary veins lah. The pulmonary vein is the blood vessel that carry the blood from the lung into the heart. So it will enter into the left atrium. After that, it will pass through the bicuspid valve, entering left ventricles. After that, left ventricle will pump all the blood through the aortic valve. So in another name is called as semilunar valve. So you can use either one. Aortic valve is also acceptable. Semilunar valve is also acceptable. So it will pass through the aortic valve, entering aorta, and aorta will pump all the blood throughout the whole body lah. And then at the same time also, when the tissue carry the oxygenated blood from the tissue region, it will go through the blood first and then pump to the lung for the gaseous exchange. So the blood, either it will go through the superior vena cava or inferior vena cava, both of them lah. Actually, you say vena cava is also already okay, but superior vena cava is located at the upper side, for inferior vena cava is located at the bottom side. They were entering the right atrium. After that, it will transfer the blood through the tricuspid valve into the right ventricle. After that, right ventricle will pump the blood through the pulmonary valve, or another name, more general term is semilunar valve, also acceptable through the pulmonary artery, and then pump to the blood. So that's all. So the most important thing that you need to know is the 4 chambers of the heart: left atrium, left ventricle, right ventricle, right atrium, and also the pulmonary veins and pulmonary artery that enter to the left atrium and right atrium respectively. And also their valve. For the left atrium and left ventricle, the valve is called a bicuspid valve. For the right atrium and right ventricles, the valve is known as tricuspid valve. Okay, and then it will pump through the blood from the left and right ventricles through the semilunar valve is more general terms. If you want to more specific, for the left ventricle is aortic valve. For the right ventricle is pulmonary valve. So yeah, but both are acceptable lah. Okay. So that one is more general structure that you already learned, and also I think this one is the size of the heart lah, meaning that the size of the heart is like the size of the clayfish. When you is about the size of the fish. This look like lah. When you compare to your hand, it is actually similar, something like that. So this is the scientific terms that you need to know how to write it. So you can say that the heart size is size of the clayfish of the adult human. Okay, so this is a location. So when you need to write about the location of the heart in human, so normally our heart is located in the chest cavity known as middle mediastinum, directly under the breastbone. So this breastbone. So this is the one of the bone name called sternum. So it is located at here lah. Actually, it's located at the left hand side. In normal person, some of the person is at the right hand side, but majority of the heart location is at the left hand side. Okay, so this is the cardiac muscle that you need to learn also about the heart. So we know that the muscle of the heart is spatial, which called as cardiac muscle, that you already learned in semester 1 well. So what is cardiac muscle? Please refer back to your semester 1, the structure of the cardiac muscle, all that actually you already learned in semester 1. So what you need to know is that what you need to know is that you need to know the muscle that cover the heart is known as, there are 3 main muscles that cover the heart, which epicardium, myocardium, and also endocardium. So this is the 3 major muscle that cover your heart. So it is actually the smooth layer of the endothelium covers the inner surface of the pericardium and the outer surface of the heart. So you can see here. So there are 2 major cardiac muscle at here, and also they have another cardiac muscle we call as pericardium that located at the inner surface of the heart, and for the myocardium and endocardium muscle is located at the outer surface of the heart. And there between these 2 surface, you can see here myocardium, endocardium, and also between here peripheral pericardium and visceral pericardium, they have the pericardial cavity. So actually this region, the space region that containing fluid that can reduce the friction to a minimum as the heartbeat. So we know that when the muscle, when your heart is functioning, have a heartbeat, actually they have the frictional force. So this body cavity is to reduce that frictional force. So this is a basic cardiac muscle structure location that you need to know. So the most important thing is these two layers, myocardium and also endocardium, are at the outer surface, and for the pericardium is at the inner surface of the heart. Yeah, so that's all. Okay, so there are also the structure we call as septum. So normally septum is at the you can see it is at the middle line that separate the right hand side and left hand side chamber of the heart. We know that left chambers and right chambers, they are totally independent. Then the blood is not mixing. The blood from the left chamber and right chamber cannot mix one. If they are mixed, they will have the meaning that this heart is have the problem. They will have the what we call as disease for the heart. But in normal situation, septum actually is the heart wall made from the connective tissue that separate the right chamber and also the right chamber and also the left chambers. So it is very important in cardiac cycle. And if you more scientifically, they have two naming of the sector, which is the inter-atrial sector that separate between atrial and intraventricular sector that between the ventricles. So the location just for atrium is at the upper side or the ventricle is at the lower side. Okay. So these actually the four major chambers that I already mentioned about the mammalian heart. Yeah. Okay. Okay, so this one I already mentioned, right? Semilunar valve. These are general terms. Actually, in examination, you can just write it semilunar valve. But if you want to write aortic valve and pulmonary valve, is also okay. But if you want to write aortic valve, you make sure they write correctly lah. If you say you write wrongly, meaning that you reverse it or you reverse the location, then no marks lah. So if you want to more be safe, don't want to be confused, is more safety that you write semilunar valve instead of aortic and pulmonary valve lah, unless you already can differentiate these two valve clearly. Okay, so this is actually the valve structure of the mammalian heart lah. If you separate the heart. So this is actually the heart. So this one is at the left hand side, bicuspid, and this one is the tricuspid. Tricuspid valve. So why it is called as tricuspid? Because of they have three lines at here. For bicuspid, they only have two lines at here. So this is their naming lah. Their naming at that. So the major function of the valve actually is to prevent the blood flow of the blood in the heart lah. Yeah, in the heart. So you need to know that in SPM, I think you already know that when the blood is coming into the heart, the blood is just flow through left atrium, left ventricle, and then it pump through the aorta. But the blood cannot go back into the atrium one. If the blood can go back to the atrium, then they have something problem for the heart. So why blood cannot go through that? Because of this valve structure. Because this valve, if this valve have the problem, something abnormal, then they will have the heart disease issue for that patient. Okay, so next is about the cardiac cycle. Okay, so before you go through the cardiac cycle, so actually what I say before is the blood flow in the heart that you already learned in SPM. So you need to know the blood flow in the heart. Then you will go through the cardiac cycle. So what is cardiac cycle? Okay, cardiac cycle. So this is actually new chapter, new topic that you never learned in SPM. So actually it is the cycle that refer to one complete heartbeat. Meaning that you know that our heart is muscle, containing cardiac muscle. So it must be able to contract and relax to pump the blood. So when the heart is contract once and contract and relax once, it is one heartbeat, right? So when you want to complete the one heartbeat, this is the situation that we call as cardiac cycle. So in just only 0.8 seconds, the heart is already complete your heartbeat. Yeah. So in cardiac cycle, meaning that they have the contract and relax, but in heart, we cannot use the terms contract and relax. We need to use the terms systole and diastole. It is very important. In examination, please don't use contract and relax terms in exam. You need to use the systole and diastole. So what does it mean? Systole is the period of contraction. Diastole is period of relaxation. Easy. Systole, contract, diastole, relax. Yeah, it is very easy. So please, again, don't use contract and relax in cardiac muscle. So normally in examination, we will use atrium systole, ventricle systole, atrium systole, or atrium diastole, ventricle diastole. Atrium systole means the atrium muscle wall is contract. Ventricle systole means the ventricular muscle is contract. Similar to this, atrium diastole means atrium muscle is relaxed. Ventricle diastole means ventricle muscle is relaxed. Just like that lah. Yeah. So this is the sequence that you must know in this topic. So this topic is very popular. So if you see this diagram, this is from the Campbell book. So actually in cardiac cycle, there are only three sequences, three events. Three events, but you here got five events. Don't need to five event, but five event. No. So don't worry, we will see it one by one. So in overall, there are only three main events only. The first event is what? The first event is when your blood go through entering the atrium first, right? At the initial condition, your blood must entering atrium first, right? Atrium first, right? So how the blood entering the atrium? Entering the atrium. So first sequence is atrial and ventricular diastole. Both of the muscle in the atrium and ventricle are relaxed. Both of them are relaxed. They do not contract one. Then the second sequence is when atrial is systole, ventricular diastole, meaning your atrium wall is contracting and ventricular is still relaxed. And then finally is that your ventricle muscle starting to contract, and then your atrium is relaxed. So this is the overall sequence about the contract and relax in the atrium and ventricles. So the first event is both of the chambers muscle are relaxed, and then after that, firstly is atrium contract first. The ventricles still relaxed, and then after that, ventricles starting to contract and the atrium back to relax, and then this cycle repeat and repeat. So this is a general condition, general sequence of the cardiac cycle. So in cardiac cycle, what you need to know? Okay, you need to know is that what happening during atrial and ventricular diastole. What happening when they are relaxed, and what happening when the atrial wall is contract, and what happening when the ventricle wall is contract. So the event that you need to know is the pressure. Of course, the pressure of the atrium and ventricles in each sequence. And the second one is the blood flow. Where is the blood flow? Blood flow from where to where? You need to know. The third sequence is the valve. The valve that I mentioned, bicuspid valve and the tricuspid valve, and also the semilunar valve is open or closed in each event. So these is the three major events that you need to know. Okay, so first of all, we need to see here. So this is the more general diagram. So if you want to go through the detailed diagram, this is from the Solomon diagram. Okay, when you go through the detail, the first event from here that is about the ultrasystole. Okay, what happening in the ultrasystole is that when the blood is already entering into the both ultra. Okay, both ultra. Okay, when the blood is already entered, so they need to empty the blood from the ultra to the ventricle first. But how the blood go through, empty the blood from the ultra to ventricle? So the ultra need to contract. So the ultra is contract. It will push the blood from atrium to ventricles. So blood is flow from atrium to ventricles. Both of them, from the left atrium to left ventricles, from the right atrium to left ventricle. Meaning that which valve are open, meaning that the tricuspid valve and bicuspid valve are open at that time. And semilunar valve closed. So this is the first sequence. So the first sequence is that atrial wall is contract. Why it want to contract? Because it need to transfer the blood from the atrium to ventricles. So as long as it contract, meaning that the blood pressure at the atrium increase, is higher than the ventricles. Not increase. You can say increase, but at the end you need to say that the blood pressure of the atrium is higher than the ventricles. And then the third event is the valve. So the valve for the bicuspid valve and tricuspid valve are open, and the semilunar valve closed. So this is the first event. Okay, after that, when you go through the after that, when the blood is already stay at the ventricles, so in normal situation that you learn in SPM is that the ventricles will pump the blood. For left ventricle, it will pump the blood through the aorta. For the right ventricle, it will pump the blood through the pulmonary artery, right? So how it pump? So what is the situation at here? So when the ventricles starting to contract, you see here, start to contract, the pressure with the ventricle starting to increase. It will start to increase. So this will shut down the bicuspid valve and tricuspid valve. Tricuspid valve. So this is very important. When the blood ventricle blood pressure of the ventricle starting to increase, it will start. This pressure will shut down the bicuspid valve and tricuspid valve as well. So meaning that the ventricle pressure now is higher than the atrium. So it must close. If these valves are not closing, when the pressure is already high like that, it will pump the blood back to the atrium. So it must close the valve. So the shutdown of the bicuspid valve, you will hear the first hard sound we call as lub sound, L-U-B. L-U-B, the lub sound, the first hard sound that you learn. You will heard at there. So this is the first lub sound produced during the ventricle systole. So why this lub sound is produced? Because of the bicuspid valve and tricuspid valve are closed. When they shut down, it will produce this kind of sound, the lub sound, L-U-B. Okay. After that, when this is at the beginning part, after that it will the ventricle will starting to increase again the pressure. When at the highest point, when the ventricles contract more, it will increase the ventricle pressure again until it is higher than the atrium. Then it will pump the blood. For the left ventricle, it will pump the blood through the aorta, right? For the right ventricle, it will pump the blood through the pulmonary artery. So at the same time, your semilunar valve is opening. Your semilunar valve are open. So in simple, what happening in ventricular systole? The first important event is the lub sound, L-U-B. Why lub sound is produced? Because your bicuspid valve and tricuspid valve are closed. Why it is closed? Because of the pressure of the ventricles starting to increase to shut down the closing of the bicuspid valve and tricuspid valve. And when the ventricle pressure increase again, it will cause the semilunar valve open so that the blood can be pumped out from the left ventricle through the aorta to whole body, and for the right ventricle, pump the blood through the pulmonary artery to the lungs. So that's all about the ventricular systole event. Okay, the final event is the aorta and ventricular diastole. Both of them are relaxing. Okay. All of them are relaxed. So when they are relaxed, meaning that what happening is that your semilunar valve must be closed already. Your semilunar valve will be closed because it already relaxed. So the blood pressure for both aorta and ventricle, especially ventricle, it will reduce. It will reduce the blood pressure of the ventricle and increase lower than the atrium one. So your semilunar valve is snapshot. So not shut down, snapshot. So similar to here. You need to use the term snapshot. So your semilunar valve is snapshot, meaning that it is closed. And when this snapshot occur, meaning that the second hard sound, which is the dub sound, D-U-B, is produced. So this is what happening. And how about the blood flow? So the airway valve was starting to open at here, and the blood will flow from the pulmonary vein for the right atrium, and for the vena cava for the right atrium, will full into the fill up the atrium, and also some of the blood will enter ventricle as well lah, as well, because the area is open, open. So at this event, you need to know is the most important thing is that the sound, the duck sound, the second heart sound is produced. Why it is produced? Because the pressure decrease, causing the snapshot of the semilunar valve, meaning the semilunar valve close. So it causes the duck sound. Sound. So at the same time, your AV valve starting to open, and the blood starting to flow from the pulmonary vein and also the vena cava into the atrium as well as the ventricles. So after that, it will repeat again. The atrium contract again, and it will repeat and repeat again. So these three sequence happening in only 0.8 seconds, approximately 0.8 seconds. And about the ultrasystole, just about 0.1, and for the ventricle systole is about 0.3. For the relaxed one is the high most time about 0.4 seconds. So that's all about the cardiac cycle that you need to know lah. So the most important thing is that I already mentioned, you need in each sequence you need to know the ventricle and ultra pressures, the open and closes of the valve, and the third one is the flow of the blood. What happened, blood flow from where to where, and you need to mention very detailed one. You need to mention blood flow for ultra to ventricles through what valve. You need to mention one. You cannot just say, oh, blood flow through valve here, but you don't mention the valve. And also you need to mention the what happened to the valve. Why it is open, why it is closed. They have the reason why. You just not say, oh, during ultrasystole that every valve open. Why it open? You need to know the reason why it open, because of pressure. Because pressure open it. And here, ventricle systole, why the AV valve is ultraventricular valve or tricuspid bicuspid valve close? Why? And when it close, it produce the sound. So you need to all relate. Okay. So that's all about the cardiac cycle. So so for those who got bought my note, actually they will have more detailed explanation at here, at there. Okay, so this is the very important graph that you must learn in STPM, and mostly it will come out in the exam. Mostly, it must come out. Especially in objective question. Normally in this chapter, they will come out. So this is the graph of the cardiac cycle. Cardiac cycle. So the most important thing is the upper one, which is the blood pressure of the graph here. So the y-axis is the blood pressure, and then the s-axis just the time lah. S-axis is the time. So you see this upper part, they have the meaning why at here, and here, and here. So you need to know there are 3 lines at there. So the upper one, this red color line, the highest line at here, the red color one is we call as aortic pressure, meaning that this pressure is measured at the aorta. And here, the blue one is the ventricular pressure, meaning that this is the pressure measured at the ventricles. And then the lowest part at here is ultra pressure, meaning that this pressure is measured at the atrium. Okay, so what happened? Okay. When we at the beginning part, the pressure is very low. You can see your pressure is low. After that, you can see that suddenly your ventricle pressure starting to increase at very, very bad. So imagine what happened at here. Why the ventricle pressure sudden increase? So you relate to the cardiac cycle. Why the ventricle pressure? When is the ventricle pressure starting to increase like this significantly? Only when ventricular systole. Meaning that ventricular systole happened from here now. So during ventricular systole, at the beginning of the ventricular systole, what happened to the valve? What valve is closed? What valve is open? At the beginning. Meaning that the airway valve is closed, right? Starting to close, meaning that the Mira valve is a bicuspid valve or tricuspid valve close. So when the ventricle pressure starting to increase, mean the AV valve close now. Okay, close. Okay, after that, when we increase until here, when we reach here, reach here, reach here, meaning what happened to here? What happened to here? You can see that not only here, the aortic pressure also increased. The pressure at the aorta also increased. Why it happened? So you can still imagine at the beginning of the ventricular systole, the pressure increase, so the airway valve close, so the closure of the airway valve, it will produce the loud sound lah, the first heart sound at here. So the first heart sound is produced at here. After that, it will increase until it reach the aortic pressure at here, meaning that at here your semilunar valve open. So here it like aortic valve lah, because this is aortic pressure, right? Aortic pressure. So meaning that you know that semilunar valve starting to open. So when the starting open, the aortic pressure also will increase because the ventricles already pump the blood go through the aorta. So it will increase significantly until after that, when it pump all the blood, blood already empty from the ventricles, it was starting to decrease. So when it decrease at here, meaning that ventricle diastole and ultra diastole starting to happen. Okay, when it's starting to happen, what is the first step first? Meaning that your semilunar valve must close first at the first step. At the first step. Because it already relaxed, meaning the pressure of the ventricles is already decreased, so it will cause the snapshot of the semilunar valve. So the semilunar valve is closed now. So here you can see at that point, your aortic valve is closed. After that, your ventricular pressure will decrease and even decrease less than ultra pressure. This is a very important part. Okay, you will see here. After that, when the ventricular pressure increase, it will similar or higher, similar to the ultra. Then it will increase higher. But at the diastole, it will decrease less than ultra pressure. So when it less than ultra pressure, it reach the ultra pressure part, meaning that your AV was starting to open. So this is the diastole region. You are starting to open. So the blood was starting to flow to fill up the ultra and the ventricles as well. So this is the everyone open. So this is the diastole state. So when the aortic valve is already closed, second heart sound is produced. So after that, it will repeat and repeat again. You can see it will repeat and repeat again. Right. Repeat and repeat again. Okay, so this is the first graph that you need to know about the graph of the cardiac cycle. This first part is very important that you need to know. Okay, this second one is the volume of the ventricles. So this one actually is not in the syllabus, but I just explaining to you lah. So actually it's very in the syllabus so you don't need to know it, but if it come out but you need to know lah. So actually at the beginning part, your volume is very high. After that, it's quite high. Why? Because at here is the diastole region, right? But at here is already blood is already filled up almost full in the ventricle, and after that it will pump in, the pressure increase, meaning that it pump empty out the blood from the ventricle. So your volume of the ventricle was starting to decrease, right? Because the pressure already increase. It want to pump out all the blood from the ventricle, and then during the diastole region, your volume starting to increase again. So this is very easy lah, not very hard one. And then the final one is the ECG, the electrocardiogram. Okay. In the ECG, what you need to learn, you need to know is that the terms we call as depolarization and repolarization. Okay, repolarization. Okay, so you see here, at the beginning part of the diastole, you can see here get the P, the ultra, the not ultra, they will have the slope line. Got the slope at there. Meaning that this part is the depolarization of the atrium occur. Yeah, you can see here because if you see here, right, you see here at the A, what you can see the ultra pressure and ventricular pressure starting to increase, starting to just increase a little bit, a little bit. This part is the ultra systole. Ultra systole is happening. So before the ultra systole, this P part is the beginning of the we call as the repolarization of the atrium, meaning that at here is the part that the ultra starting to do systole. So this is we call as depolarization of the atrium. Depolarization of the atrium. Then you will see the QRS region. It will like something look like suddenly decrease, suddenly increase, all these. This is the region we known as the depolarization of the ventricle. Right. Because you can see here when the ventricle starting to increase, all that, this is a region that it is the depolarization of the ventricles. And then after that, you go to the T. After that, after QRS, you will have the T part. T1, T1, T1. This is the region correspond to the repolarization of the ventricles. QRS is depolarization and the T is repolarization of the ventricles. So yeah, the just that you need to know lah. You need to know lah. Yeah. Yeah. Okay. So that's all about the graph or cardiac cycle that you need to know lah. Okay. Okay, next we go through the topic about the heartbeat initiation. Okay, so this topic actually you also learn in SPM as well lah, as well how of heartbeat is occur, because we know that heart is myogenic, right? Because it is involuntary action one. When you are alive, your heartbeat is still happen. Only the people is already dying does not have any heartbeat. Even you are sleeping, you still have heartbeat, right? So if you still have the heartbeat, so how what signal trigger the heartbeat to occur? Because you still remember that you learned in SPM about the nervous system, you need to have an electrical signal one to trigger the muscle to contract and relax. So why you can walk, why you can take the things, why you can see anything, why you can run, you can do exercising, all these is the motor neuron, right? You need to have a signal. So in heart, what is the signal that trigger your heartbeat to contract and relax to do all that target cycle that you learned just now? Okay. So this is the general mechanism that you need to know, right? All these you need to know. So SA node and AV node, all you already know, right? What is SA node, what is AV node, sino atrial node, and also ultraventricular node, ventricular node. So this actually just abbreviation about in your SPM because all of these you already learn in SPM. Okay. First of all, we know that sino-atrial node is act as the pacemaker and located normally at the right atrium at here, right atrium, right atrium. Okay, when your SA node is already starting to act as a pacemaker, it was automatically generate the electrical impulse one, electrical impulse. So when it generate the electrical impulse, it will send the signal to the atrium first. Send the signal to the atrium, causing the atrium to contract. After that, they will have another node we call as ultraventricular node. The ultraventricular node will receive the signal from the SA node, and it will be stimulated, and will starting to send the signal through the bundle of branch, and then go through the Purkinje fiber and pass through the Purkinje fiber and conduct the impulse to both muscle fiber of both ventricles and make them to contract. Right. So this is a general that you already know lah. But the more detailed thing is that before the AV node send the impulse through the bundle of branch to the Purkinje fibers, it will delay the signal is delayed first because it need to ensure that the blood from the atrium go through the ventricle. So meaning that when the SA node generate electrical impulse to the atrium, it will stimulate AV node, but the AV node will not directly send the impulse first. It will delay the signal first about the 0.1 second to allow the blood from the aorta go through the ventricles. So the ventricle signal will go through the bundle of bar Purkinje fiber until it reach the heart apex throughout the ventricles, and once it receive the signal, and it will starting to the contraction of the ventricles and pump the blood to empty out the ventricle. So that's all about the heartbeat initiation. Okay, next we go through the regulation of heartbeat. So we know that heartbeat is initiated by all that mechanism before is sinoatrial node and ultraventricular node. So when you're doing exercise, you will feel that your heartbeat is increasing, right? And when you feel cold, at the very cold region, your heartbeat will reduce. So what regulate this heartbeat? So one of the major region is the nervous system. Of course, your nervous system still can control your heart rate one. So how it control the heart rate, it will go through the sympathetic and parasympathetic nerve system. So these two nerve, you will learn more detail on the next chapter chapter 10, control and regulation. There will one topic about the sympathetic nerve and parasympathetic nerve, and you need to know their differences. So in this chapter, you just want to know that what are the role of the sympathetic and parasympathetic system that influence your heart rate. Of course, adrenal gland also will secrete the two hormone, adrenaline and noradrenaline. Epinephrine is adrenaline and norepinephrine is noradrenaline. Epinephrine is the name that they use in US. In British, they use the adrenaline. So these two hormone, we know that it is for the fight and flight action, and it will increase your heartbeat lah. Okay, heartbeat lah. Okay. Okay, so next you need to know about the parasympathetic nerve effect. So in parasympathetic, you need to know in parasympathetic, the neurotransmitter that it release is acetylcholine. Okay, what is neurotransmitter? Okay, although you still not entering the next chapter yet, but I think in SPM you already learned about what is synapse, right? And the nerve impulse transfer from the synapse, from the nerve entering into the synapse, right? This one you already learned in SPM. So you can see here is actually the nerve, the synapse, one of the synapse structure lah. But actually this one is not a synapse. It is what we call as neuromuscular junction that you will learn in next chapter. So for now, if you still confusing what I'm talking about, you can refer to my next chapter video lah. But it's okay for now. You just want to know that in the when for the parasympathetic nerve effect, it will release acetylcholine. Okay, what is the role for the acetylcholine at the muscle? At the muscle. So it will bind to the acetylcholine receptor at the muscle membrane, and it will activate the G protein, and this G protein will bind to the potassium channel that at the plasma membrane, and initially it is closed and it will open the potassium channel. So when the potassium channel is opening, meaning that the potassium ion from the inside the cell will release out, causing the hyperpolarizing, meaning that the action potential is occur more slowly. Okay. For here, you will be very confusing. What is action potential? Okay. For this topic, this part, I recommend you after you learn the next chapter, then you refer back to this chapter, then you will know more accurately. For now, maybe you are very blur what I'm talking about. So this part, I recommend you learn from the next chapter first, then after that you revise again, then you will very know what I'm talking about. What is action potential? Why potassium ion release out, it will cause the action potential reduce. So action potential in general term is the activating of the nerve impulse, meaning that when action potential occur, contraction will be occur. In simple way. So meaning that when the increasing if it say that action potential occur more slowly, meaning that your contraction of the heart is reduced, right? It already reduce and reduce. So acetylcholine is play the major role to reduce the heartbeat rate. So E is for the parasympathetic system. When the parasympathetic nerve is activated, it will reduce the heart rate. So that is why the E is red color, decrease. Well, another one, sympathetic is noradrenaline here. Noradrenaline. When it will release the noradrenaline for the sympathetic nerve effect. Okay, so this noradrenaline will bind to the receptor on the plasma membrane of the cardiac muscle, and this receptor will activate the G protein again, and this G protein will activate the CAMP reaction, and finally, the most important thing is here. Protein kinase will phosphorylate the calcium ion channel so that it can open easily. So more calcium ion will entering into the cell so that the action potential is more rapidly occur. Okay, so again, this topic you also need to require to learn about that chapter, then you will know more detail about it. What is CAMP? So actually CAMP is one of the mechanism in the non-steroid hormone, non-steroid hormone. And calcium ion is very important in the neuromuscular junction mentioned one. So in neuromuscular junction, you want to contract, make your muscle contract. Calcium ion is one of the play major important role for muscle to contract. So when there are more higher calcium ion at the inside your muscle, meaning that your action potential is very easily to occur. So in simple word, for the sympathetic nerve effect, it will release the neurotransmitter we call as noradrenaline. So noradrenaline will increase the heartbeat rate. So for the acetylcholine reduce the heartbeat. For noradrenaline, it increase the heartbeat. And another one is the adrenal gland. So adrenaline and noradrenaline lah. They will secrete these two hormones. So these two hormones play the major role in flight and fight response that you learn in SPM1. This one topic is you learn in SPM1. So when the person respond to this flight and fight, of course it need to increase your heartbeat rate lah. Okay, the next chapter, next topic is about the cardiovascular disease. Okay, so in our syllabus, the first terms that you need to learn is arteriosclerosis. Okay, please ensure this is arteriosclerosis. There are more general terms that you heard mostly is atherosclerosis. These two terms are different. Please be alert. Don't confuse it. Please don't say that arteriosclerosis is same as atherosclerosis. No. There are different definition. So because in syllabus they already separate these two terms. So of course these two terms are different. If they are similar, they just put the bracket one. So this is the definition of the arteriosclerosis. So meaning that it is a condition occur when the artery walls become hard and stiff that can already interfere the blood flow or disrupt the normal working of your circulatory system. So the most important thing is hard and stiff. It already make your artery wall become hard and stiff. And in general, there are three types of the arteriosclerosis lah. So arteriosclerosis, arteriolosclerosis, and this one I don't know how to read. Yeah, but you don't need to know all these. You just want to know arteriosclerosis. This is a major one. I think in SPA you also learn it. Arterial. Yeah, these two you don't need to learn. You don't need to know. You don't need to know. You don't need to know it, because this one is like a thickening of the arterials wall one, because it already say like arteriolosclerosis, meaning that it is the part that occur, the thickening of the artery is occur at the arterials vessels. Because you know, like artery is more bigger, the volume, the diameter is more bigger, and they have the more smaller one is arterioles. So this one is the disease that occur at there. And another one, this one is the is the calcification process, because it already say that calcify, meaning that the blood arteries is already be calcified, become hard lah, become hard lah because of the accumulation of the calcium ion. So these two you don't need to learn it, you just learn about arteriosclerosis is already okay. Okay, so what is arteriosclerosis? So arteriosclerosis actually is the disease where your large interior wall of the arteries is already damaged, inflamed, narrow, and as a result of the lipid deposit in their heart wall. So this one is actually more detailed out for the definition. So for arteriosclerosis, it's just natural term only. They just say that the artery wall is hard and stiff enough. They have no plug at all, no blood, they don't have blood at there. You just say that, and the blood artery is not narrowing, no narrowing one. For the definition of arteriosclerosis, there are no blood, and the artery lumen is not narrowing. So this is a definition of arterial. For arterial, it is where your internal wall is already damaged, inflamed and narrow. Narrow is very important. Narrow is important. And what it causes it narrow because of lipid deposit at your artery walls. Here. So you can see here, this one. This is the plug. And actually, this one is also another plug we call as thrombus. So actually, in the normal situation, your blood need to look at here. All these should be white color one. We call as lumen. Or you need to be white color one so that your blood can pass through, right? You can imagine your blood versus artery is like a pipe. But you can see here, your pipe is already block all these things. You can imagine one. You can imagine when your pipe is all block all these. Can you imagine your blood can easily to flow through this part or not? Cannot, because the lumen is already become narrowing. So this is the arteriosclerosis. So what you need to learn about this chapter, you need to know how this plug is formed. You just not just say that effect of the arteriosclerosis only. Because in SPA, you learn more detail is about effect of arteriosclerosis. What is the effect? And then the general causes of it. We know that all these is because of you do not have the healthy diet, eat too much of the junk food, all the fat things. This one is SPA level. You can say that this. But in STPM, they will ask you to write out the development of the arteriosclerosis. So how, and the marks normally is very high, 9 marks, 10 marks. Can you say like, because arteriosclerosis happen because of you drink too much of salt water, do not have the healthy diet, do not do exercise? You write like this in STPM, no marks at all to give you. You need to very write in scientific terms one. So how this happen? How this happen? Okay, first of all, you need to know is that the endothelial layer is damaged. Okay, what is endothelial layer? Okay, endothelial layer will refer back to your structure of the artery that you already learned in SPM, meaning is that the surface layer of the artery wall. Not surface layer, inner layer of your arteries, meaning at here lah. At here. This is artery, right? This is artery, and at the inner layer, is we call as the endothelial layer lah, endothelial cells. Okay, so normally when there are the fat deposit, normally is LDL cholesterol lah, LDL cholesterol or calcium, eat too much of salt, they will accumulate at here. At here, or accumulate at here. You can see here, it will accumulate at here. Okay, once it accumulate at here, actually, these are all the foreign substance. They are foreign substance one. It will trigger higher amount of macrophage to settle this problem. Because in general condition, this part it should be empty. They do not have anything one, but they have anything. They have things. So meaning that the macrophage need your immune system need to be activated to settle this thing. But actually, this thing is not a pathogen. It's not bacteria. It's just a fact that already cannot already cannot flow already. They just stick at there because stick at that. But your immune system don't know what. They don't know this is bacteria or what But they assume these are the bad things that need to be settled. So when the macrophage starting to do this, we know that macrophage will play the role in phagocytosis, in immune system, right? Okay. So it will aggregate at here and eat the fat. Eat the fat, but it cannot digest the fat because the macrophage major thing is to digest the pathogen, digest the bacteria, digest the virus, digest the antigen, not digest the fat. So when it eat the fat, it cannot digest it, and at the end, the macrophage die. It will cause the macrophage die. Die. And once it die, it will accumulate here again, and it will trigger another macrophage, eat that kind of macrophage again, and then it cannot digest it, and it will repeat and repeat, meaning that it is already causing inflammation. Because when there are many white blood cells aggregated here, meaning that inflammation is already occur. But the macrophage cannot settle this kind of thing because they are not bacteria. They cannot digest the fat one. So when it aggregate at there, it will starting to form the foam cells. FOAMCELL. So after foam cell, here, you can see here like this fatty streak formation, like here, foam cells like this. Foam cell is where the macrophage start eat the fat, but they cannot digest it, and it will eventually die. So this die macrophage is called as foam cell. And because they already maintain at there, your smooth muscle tissue of your artery was starting to migrate to the inner lining wall of the artery and multiply along with the connective tissue, and other ECM substance also will proliferate and produce collagen here. So because of this part of this thing accumulation at there, so your smooth muscle tissue muscle tissue need to aggregate at there and cover all this, because they think that why your smooth muscle tissue actually at here, but the cell why suddenly got something at here? No, they should not. Something at here, because smooth muscle tissue, the label one it should be endothelial layer. So the smooth muscle tissue starting to reproduce at there and starting to form something like this. Yeah, you can see here. Your smooth muscle tissue starting to cover all the foam cells at here. Starting to cover all the foam cells. So this environment actually is already bad environment, already trigger inflammation. This environment will also trigger more and more macrophage aggregate at here, and more foam cells form at here. So actually this is a very bad recycle process. So this all the accumulation, when you all accumulate all the lymphocyte and macrophage and those smooth muscle tissue all accumulate here, it will form the lipid blood. Of course, it will form the lipid blood. It will form the blood like this. So this already is considered as a blood already. Not a normal situation, it already become a blood. Because in the inner part of this, all are lipid. All are the lipid, because the smooth muscle tissue starting to cover all these things. So your smooth muscle fibers starting to synthesize a protective fibroscape and sequestering or isolate the blood from the arterial lumen and circulating the blood. Because the smooth muscle tissue need to cover all these to make sure the blood can be passed through. This is the origin of the smooth muscle tissue want to cover it, but actually is making the situation more worse. But he don't know. The smooth muscle don't know one, because it need to cover it, ensure that the white blood cell can fight at here, and ensure the blood flow is more smooth. But actually it makes the situation more worse. So when the blood is very thick, thick, it will cause the formation of the thrombus thrombus meaning that in the first situation, your blood actually is still stick at the lumen wall, wall, but lumen wall. So at here, autoscosis is already happening. Your lumen of the wall is already become narrowing, and your wall of the artery become hard and stiff. All that already become all hard situation at there. And when your blood is become bigger and bigger again, it will trigger the process we call as thrombosis, meaning that this blood will be released out, not stick again. It will be rupture. We call as rupture, meaning that this blood already released out from the wall. It will not stick anymore, and it can be flow anywhere through the arteries wall, through the blood, because the blood is water, right? One type of water, not water, is liquid. So it can take this blood go through to anywhere, and when this blood stick to the arteries that cannot move anymore, then it will cause the thrombus. So when the blood is already released from the rupture from the artery wall, this we call as thrombus. And these thrombus can completely block the flow of the artery. When it go when it maybe in example, maybe it flow through, it is rupture from the 10 mm cube of the 10 mm cube of the diameter. When it reach to the 5 mm cube, then this will block to that artery, and then no blood go through at there, and you will cause the very dangerous situation, because when the blood cannot pass through it, meaning that at that tissue region maybe will have some problem at there. Okay, so this is a blood rupture. So when the blood is rupture, is released from the wall, it will cause the thrombosis process and cause the thrombus. Okay, so this is a diagram more detail. This is a blood, right? This is a blood. Blood. And when it is starting to release out, rupture it, the thrombus will be formed at here. And this thrombus can go through at anywhere one until it block at there, some place, and it cannot move anymore. Then it will block the artery wall, not artery lumen, so that the blood cannot pass through it, and this will cause very serious and dangerous. So this is a normal one, healthy person. This is an endothelial layer, inner layer at here. And when there are the very many lipid deposit at here, accumulate at here, what happens is that the macrophage starting to engulf all these things, but the macrophage is fail to digest all these things actually, and it will trigger more and more lymphocyte or macrophage start entering into it, and the small muscle tissue starting to aggregate that area and starting to form the new layer of the muscle tissue, and this one blood is formed. This is the blood. And when the blood is bigger and bigger, it will rupture out to form the thrombus. That's all about the development of atherosclerosis. Okay, so this process is, I just use the diagram. For those who got bored might not, you will get the more detailed explanation, scientific explanation. This is just a general process. Okay, so after this is the effect of the atherosclerosis. Atherosclerosis still happening, not effect, major cause of the cardiovascular disease. So one of the signal that you have the cardiovascular disease or not is the hypertension, high blood pressure. So just that you need to know the normal blood pressure lah. In normal blood pressure is actually at the 120 over 80 mmHg lah, 120 systolic and 80 diastolic blood pressure. And if you normally is above 140 and /90, you are already considered as the hypertension. Normally is already considered hypertension. Yeah. Okay, so how hypertension relate to the cardiovascular disease? So normally you need to know the definition of the blood pressure first. So blood pressure. So when you can imagine when your arteries become narrow or stiff or have something blocked at there, actually you are forcing the heart to pump more pressure. You need to make your heart work more and more. Because the blood is cannot flow smoothly. So something like in the highway lah. If you drive in highway, you are very smooth, right? No car. In the highway there are no car at all. But when you drive in like a kampong road or culture road, they have many car, many traffic light at there, so that it is very not smooth. So you need to have a high energy at there. So your heart need to pump more and more energy, increase your pressure to maintain the flow so that every part of your cell can receive the blood. So this over time, this overwork can weaken your heart muscle lah. Yeah. So this how hypertension relate to the cardiovascular. And next is about the heart attack. One of the heart attack terms it be called as myocardial infarction. Is one of the common name is heart attack. So myocardial infarction is one type of the heart attack. Okay. Okay, so normally this is happening when your coronary artery of your blood coronary artery is blocked. Okay, what is coronary artery? So normally coronary artery is the blood vessel that carry blood to the heart. You need to know the heart still is the cell. It is the organ. It's still a cell. It containing cardiac muscle. Cardiac muscle is still the cell. They still require all the general cellular respiration, glycolysis, all that, Krebs cycle, etc. They still energy one. You need to pump. They still energy. They are not the dead cell. They still are the cell. So they still require oxygen. They still remove the carbon dioxide as well. So coronary artery is the blood carry the oxygenated blood to the heart so that the cardiac muscle can function normally. But in heart attack of the myocardial infarction, meaning that the coronary artery is already blocked. So what happened to your heart? When your heart cannot receive any oxygen anymore, do you think your heart can function? Cannot. So this is very dangerous situation of the one of the heart attack when your coronary artery is blocked. But if it is partially blocked, it will cause the condition we know as the angina pectoris. Angina pectoris. But when it is totally blocked, it will cause myocardial infarction. It is very dangerous because your cardiac muscle is already starting to die. If partially blocked, meaning that the oxygenated blood still can pass through, still can enter it, but the amount of the oxygenated blood is very low already. This one actually is already can starting to damage your heart and you will cause the heart pain already. But if for the myocardial infarction, it's more very serious, and normally if you not do the CPR immediately, normally these people will die in few minutes. Yeah, so this is very dangerous. Okay, so of course, causes of the cardiovascular disease, high sodium intake, happening, high sodium intake, and also the LDL cholesterol. What is LDL cholesterol? Please refer back to your chapter 1, semester 1, chapter 1 as well. LDL cholesterol, HDL cholesterol, and all that. Of course, smoking containing carbon monoxide and also nicotine. They can affect your blood vessel, your blood artery. Finally, obesity. When your BMI is higher than 30, and your body weight, all that, obesity associated between the blood pressure, blood lipid, blood glucose, all is very high. Your cholesterol level is very high, blood lipid is very high, and adipose tissue, all this is increasing. All this, yeah. So another one is stress. Even though some people look like very healthy, but they still have heart attack as well. Why? Heart attack and atherosclerosis as well. Why? Because of stress. Because stress hormone like cortisol, it was starting to hydrolyze glycogen in your liver. So when your glycogen is hydrolyzed, meaning that high, that your glucose concentration is very high. So it will increase your blood cholesterol, blood lipid, blood sugar, and your blood pressure lah. So when you have many hormone like cortisol like this, and in long term, it will cause many effects. And also the hormones starting like adrenaline, noradrenaline, that I mentioned before, right? Adrenaline, noradrenaline, it will increase your heartbeat rate. Heartbeat rate. Okay. 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 So one of the prevention in the cardiovascular disease, of course, these are all the prevention but advantage of the prevention is that exercising. Doing exercise is the best prevention for you. Exercising, take a gym, do a jogging, or exercising 1 week per three times per week, and these are all the advantages why exercising is very good. And this is data about the exercising. When you do the low intensity of exercising, and the frequency time, you will release out how many kilocalorie. And this walking and differences between walking and running. When you doing a running, meaning that running is more better than you do the walking as well. And you do, and of course the diet, healthy and balanced diet. You need to less eating the hot drink, the high sugar, high fat, high salty. You should eat more the vegetable, eat more original food. The most thing is the original food, not additional food, not the processing food, not hot dog, not ham, not sausage. All that is the processing food. You should avoid that. You should eat the original food as well. Okay. The next one is the lymphatic system. Okay, so in lymphatic system, actually in this semester, there are 2 chapters you will learn about lymphatic system. So this chapter, lymphatic system, is talking about the transport system. And another chapter is the chapter 13 or 14, chapter immunity one. Okay, that chapter, lymphatic system, is more focusing on the immune system. Okay, so in this chapter, lymphatic system is more about to transport. It is one type of the transport system. So this is a function of the lymphatic system. So we will more focusing on the collect and return the interstitial fluid to the blood for this first function. The second function and the second function is at the chapter immunity, and the third function actually is not our part lah. This one is actually you learn in SPM lah, because in STPM there are no nutrition topic. So this third function actually is not in our syllabus. So we will focus on the first function. So of course this one you already learned in SPM lah, interstitial fluid formation. How the interstitial fluid is formed. So we know that at the arteries and wall between the arteries and wall, they have the blood capillaries, right? And at the blood capillary region, then will have the formation of the interstitial fluid. So it is actually exchange of the substance between the blood and interstitial fluid take place across the thin endothelial wall of the capillaries. Okay, so you can imagine that when the blood is go through the arteriole, the most important term is that the high hydrostatic pressure. Your hydrostatic pressure is higher than the venule part, right? So your sum of the plasma of the blood is forced out from the capillaries into the tissues region, which is the interstitial space at here. So once it leaves the blood vessel, the fluid is called as interstitial fluid. So normally, the most important thing is in STPM is this. All that just now I stated, actually you already learned in SPM lah. But the most important thing is this. You need to know about the osmotic pressure and the hydrostatic pressure. Okay. So in general, when the blood is enter to the arteriole and arterioles go through the venule part, it will pass through the blood capillaries part, right? And blood capillary is the region that region that formation of the interstitial fluid. Okay, so you need to know about the relationship between hydrostatic pressure and osmotic pressure. Or osmotic pressure. So at the ulterior end, your blood pressure is very high. Hydrostatic pressure is high. Is high. But your osmotic pressure in the blood actually is an opposing force acting to draw the fluid into the blood. So what is the difference between the hydrostatic pressure and osmotic pressure? Osmotic pressure is the pressure that produced by the difference in in the solute concentration across the plasma membrane. So it is a measure that can be measured pressure that can be measured when there are the solute concentration difference. So you can imagine that when the arterial pressure is entering, your hydrostatic pressure is confirmed is already very high. But your osmotic pressure will force acting to draw the fluid into the blood. So meaning that hydrostatic pressure is the pressure that play the role to forcing out the plasma from the blood capillary into the interstitial fluid. Osmotic pressure is the pressure that draw the interstitial fluid into the blood capillary. So in general terms, hydrostatic pressure of the blood generally is higher than the osmotic pressure of the blood one. So osmotic pressure is low. And there are also the osmotic pressure at the interstitial fluid. So when the osmotic pressure in the interstitial fluid is very high, meaning that it can extract more and more blood plasma from the blood capillaries. So in simple terms, if you want to force out the blood plasma coming out from the blood capillary, hydrostatic pressure must be higher than the osmotic pressure of plasma and low and higher than the osmotic pressure of the interstitial. So this is what we call how the calculation happen. When we want to calculation whether this is the filtration or adsorption. Filtration is the process that the blood plasma is already coming out from the blood capillary. Adsorption is the process where your interstitial fluid coming back to the blood capillary. This is a calculation. So you add up the hydrostatic pressure, add up with the interstitial osmotic pressure of the interstitial fluid, and minus the osmotic pressure. You will get the either is positive or negative. If it is positive, meaning that this is a filtration pressure. Why? Because the pressure is higher than outside. The pressure in the blood is higher than outside. But in the venue end, when it is at starting to reach that venue end, your hydrostatic pressure is already reduced, but your osmotic pressure may be increased, and osmotic pressure of the interstitial fluid may be increased, may be constant. For this situation, it assume it is constant, but actually in real situation, it will change. So similarly, you just add up hydrostatic pressure, add up with osmotic pressure, and minus the osmotic pressure in the plasma, you will get the negative value. If you get the negative value, meaning it is adsorption process. So meaning that the interstitial fluid at here will be adsorbed by the blood capillary. This is the interstitial fluid formation between the hydrostatic pressure and osmotic pressure relationship. So so this is a calculation that you need to know how to calculate it, because normally it will come out in objective question. So this is okay. So once interstitial fluid is formed, you can see here, once the interstitial fluid is already formed through the net filtration process, so this interstitial fluid will go to the lymphatic system. So it will diffuse into the lymph capillaries, and finally it will go through the lymphatic vessel, and it will pass through the lymph node. As the lymph circulate through the lymph node, phagocyte will filter out the bacteria and other harmful material. So this is one of the lymph node function. So if you want to know more about lymph node, you will learn more about in chapter immunity. Okay, so the most important thing is that all the lymphatic vessels finally will join to the circulatory system, the blood circulatory system, at the base of the subcapital veins by the way of the duct. thoracic duct and the left hand side and right hand side of the duct. So more easy lah. So you can see here, this is all about the lymphatic vessel. And finally, all when you go through the lymph node, you can see here there are many. You can see there are many lobes at there. This is a lymph node. Finally, the lymph will go back into the blood circulatory system through this subclavian vein. Through this subclavian vein. So either it is through right subclavian vein or left subclavian vein, and also the lymphatic duct. Yeah. So if you want to know more detail about explanation, you can book my notes. Yeah. Okay. So this is the abnormal functionless of the lymphatic system. So one of the example. So if the movement of the lymph in lymphatic system is disrupted, will result in the fluid accumulation, will causing the oedemia. Which you can see here her leg lah. Normally the main or the symptom is their leg. The leg is very big one. Okay. And one of the reason is the parasitic worm. When your parasitic worm entering into the lymphatic system and your immunity cannot against this parasitic worm, then it will cause this oedema process. Okay, so that's all about the human part. Next, we will go through the plant part, the vascular plant part. When we talk about the transport in the plants, so normally there are two major processes. If you get in your SPM syllabus, the first one is transpiration and the second one is translocation. And then the third one is root pressure. So similarity, in STPM, you still learn these three processes: root pressure, transpiration, and translocation as well. Okay, so when you learn about these, so of course, I need you to recap about your Sem 1, Chapter 3, about the vascular tissues, xylem tissue and phloem tissues. So you will still remember that in xylem, they have four major types of cell, right? And xylem vessel, tracheic fiber, and parenchyma. So the most important thing is xylem vessel, because xylem vessel play major role to transporting water and mineral ion, right? Right. So about the structure of xylem vessel, all these, although it is Sem 1, but you still need to know what about it. And also the location here. The location. You still need to know because in objective question normally maybe it will come out something question like that. Okay. So water transport in plant. Okay. So normally we know that when the plant need to absorb the water, it normally go through the root cells. Before it entering the root xylem, it need to absorb the water by the root cells. So the water you can see here, this is a root hair. The water is absorbed and then it will enter into the xylem vessels in the root. So how the water is passed through from here, from the root hair into the xylem vessel. So this is the process that you need to learn. Okay. So there are three major processes that you need to learn about that, which is the apoplastic pathway, symplastic pathway, and vacuolar pathway. Not transmembrane. These three: symplas pathway, apoplast pathway, and vacuolar pathway. Only these three pathway only. Okay. About symplas pathway, what is happening in symplas pathway? So symplas pathway, normally your water is transported through the cytoplasm. Here you can see the water is through the cytoplasm. And how about when the water want to transfer from one cell to another cell? So it's through the cell wall. So it's through the cell wall of the plasmodesmata. So that it can transfer the water from one cell to another cell, and finally it enter the root xylem. Another one is vacuolar pathway. Okay, so about the vacuolar pathway, already mentioned it is vacuolar. So the water is transferred from one vacuole to another vacuole by osmosis process. Osmosis process through the cytoplasm. Yeah. So when it enter the vacuole, and then it will go through the and this water will directly enter to the another cell of the vacuole through the cytoplasm. Okay. Another one is about the apoplast pathway. Okay, so apopus pathway is only the water is passed through the cell wall only. It will not entering cytoplasm, it will not entering vacuole. It only go through the cell wall only. So yeah, that's all about the three major pathway. But you need to alert that in the apopus pathway, the water will not directly go through the xylem vessel. For the apopus pathway and vacuolar pathway, the water can be flow from directly from the root hair, from one cell to another cell, directly enter the xylem vessel. But for the apopus pathway, when they reach the endodermis layer at here, you can see this red color one. This is the last region before it entering the xylem vessel. When it entering here, meaning that the water cannot pass through this endodermis layer. Why? For apopus, only for apopus. Why? Because they're containing the containing the structure we call as Casparian strip. This Casparian strip. Because this Casparian strip is made up of the wax we call as suberin and impermeable to water at the cell wall. Now, so you can see here, because apopus pathway is the water move through the cell wall, but until it reach here, the water cannot pass through already because of this structure, this Casparian strip. So, because of this pressure of the Casparian strip, so it is be forced that the water move out from the cell wall and either entering simplest pathway or vacuolar pathway only. So this is the key reason that you need to know. Need to know. Yeah. Okay. Next is about the water movement from the roots to the leaf. Okay, how the water is moved from the root to the leaf. So through these three major processes: root pressure, cohesion, tension theory, and transpiration pool. So all these three you already learned in SPM, right? These three, but in SPM you just learn the surface only. So in STPM you will learn very detailed. Okay. So in root pressure, so in SPM normally you say that root pressure is the first event. But actually, root pressure is happening. Normally, there are no transpiration process. So, meaning that it normally often occur at night one. At night one. So, during the day, actually, root pressure is not occur. Root pressure normally is occur at night. At night, it is the pressure that exerted by water in the root in response to a solute potential. Okay. So, in generally, when your water is moved into the root is pushed up through xylem vessel to the top of the plant. So it is a pushing force. Compared to the transpiration is pulling force. This is the pushing force. Okay. So how the root pressure occur? So when you see there, mineral ion normally is actively absorbed from the soil are pumped into the xylem vessel. So they are active transport process. Okay, meaning that we will see this diagram when the water is at water at there. When you want to root hair want to absorb the water at the water at the outside region must be higher than the inside region, right? So the water can be diffused into the cell through the osmosis process down the concentration gradient. Okay, so for the root pressure, the mineral ion, it is solute potential solute concentration, solute is actively pumped into the xylem vessel. So instead of water, because instead of water, it is using the mineral ion actively pump into the xylem vessel. So what happened to your xylem vessel? Your xylem vessel solute concentration increase, water concentration decrease, right? So when your water concentration decrease, meaning that the water from here, from the endodermis part, can start to diffuse into the root xylem vessel. So it provide the osmotic effect. Osmotic effect. So accumulation of the osmotic effect. So the when the water is accumulated in the root tissue, so that it is high water potential, so the water will push up to the plant up to the plant down the concentration gradient through into the xylem, into the shoot xylem. Okay, so because of this pushing force, so normally root pressure is not very strong one. Because you need to know we have gravitation. You use gravitation force. So if you just use the root pressure pushing force based on the water potential osmosis theory only, actually is not very strong. So root pressure is not strong enough to explain the rise of the water to the top of the tall trees. So if it is a short tree, of course, it can go from the bottom to the top. But if it is a tall tree like 10 meter, 20 meter, even 100 meters, woody plant like that in forest, root pressure is not strong. So normally root pressure is just for the plant to take the water at night. Okay. Okay, so root pressure will cause the glutation. So this is a phenomenon where the water is forced out through the spatial opening in the leaf called hydroton. So that is why that when you wake up in the early in the morning, you go to see the plants, small plant, you will see the leaf have the water at their leaf surface one. This is what you call as glutation process. And this glutation process is caused by the root pressure. Yeah. Okay. So this is about the root pressure. Okay, so the most important thing is about the cohesion and tension theory. So normally cohesion tension theory is happening at the shoot xylem of the plant. Okay, so theory. Okay, so this I need to mention that cohesion tension theory is that you use the cohesion force and adhesion force of the water water. So water, and it is a pulling process of the water from the root into the top of the plant. So you need to know how water can be pulled out from the roots into the top of the plant. How? Okay. So in general, the xylem sub is water in the xylem. Normally, it has the negative pressure. It have the negative pressure. So if you want to learn about the cohesion tension theory, you need to learn about see this picture first. Okay. During the day, what happened to the plant? If you still remember that your stomata is open, right? Your stomata is open. During the day, your stomata is open. Okay. So when your stomata is open, what happened to your plant? Your plant will do the gas exchange, right? Gas exchange, carbon dioxide and oxygen is moving in and out. But at the same time, transpiration also occur. So meaning that when your stomata is opening, your water vapor at the leaves is released out from the stomata. You can see here, diffuse from the moist air space of the leaf to the drier air. Why? Because the atmosphere has low water potential. It is more drier. So water is very simple. The theory movement of the water is very simple. It will flow from high water potential to low water potential area. So atmosphere is low water potential area. So the inside of the leaf region is high potential. Outside of the leaf is low. So all the water from the leaf will move out. So what are the water? The water vapor at the leaf was starting to release out through the stomata. Okay. So when you see the diagram structure of the leaf, this is the diagram structure of the leaf. In the mesophyll region, there are many air space at here. You can see. There are many air space, meaning that there are many water vapor at here actually. Okay. When you go through the xylem at here, mesophyll cell, xylem cell at here, there are many mesophyll cell at here. Yeah, right. And normally your mesophyll cell is filled up with all the water like this. At first, your water vapor lost by transpiration is replaced by evaporation from the water film that coats with mesosphere cell. Okay, so I just need to very simple that mesosphere cell in the mesosphere region, mesosphere cell actually is arranged loosely. There are many A space at there, and along the mesosphere cell there are many water caught at there. Only and you can imagine that you can imagine that at the cell wall of the mesosphere cell there are actually many water are caught at the surface of the cell wall one. There are many water at there. Just like the layer of the water, you can imagine there are many layer of the water at there. This is what we call as the water film that caught with mesosphere cell. This is a water film, water film that caught with mesosphere cell. This is a mesosphere cell when you enlarge the cell wall of the mesosphere cell. Okay. As I mentioned, this is a water vapor at the air space at the mesosphere cell. So when the water vapor is already released out, so that the air space does not have any water vapor. The water vapor in the air space reduce. So in plant, you need to maintain the moisture of the air space in the mesosphere cell, so that the water vapor lost by respiration is replaced by evaporation from water film. So this water film actually is caught with the mesosphere cell one. But since the water vapor is already lost, so it was starting to evaporate to produce more and more water vapor to replace the loss of water vapor. Water vapor. After that, this will cause the water film become curvage, more curvage. And this curvature will increase the surface tension of the water and the rate of transpiration in the mesosphere cell, in the plant. Meaning that the shape of the water film become more curved and the surface tension increase. So the rate of transpiration starting to increase again. Okay, so this will cause the pulling force to belong to the mesosphere cell until it reach the xylem vessel. This is one of the mesosphere cell. So the water cord of the mesosphere cell is already released. You can imagine that the water film already no water. So I need to replace this water. So this water, how it replaced? Where is this water come from? So the increased tension will pull the water from surrounding cell and air space. So if you want to maintain the water first at the water form straight, you need to take the water from other places. So how other places? So the other places is surrounding cells. So it will take another water. If there are any water from the airspace or the surrounding cell, it will take to replace the water film, so that it can become the original water film. Okay, so this is the pulling force. So this will produce a pull tension that pull the water out from the leaf xylem toward the mesosphere cells. So you can imagine like this. Mesosphere cell release the water already. It already used up the water. So the cell alone already don't have water. So I need water. Where I can get the water? Either I get from my friends. Ini kawan-kawan saya. I get semua kawan saya. Okay, saya dapat air, saya tak ada air. Tapi semua kawan-kawan pun nak air juga. So macam mana nak replace all these? So finally it will get the xylem vessel. Because xylem vessel have many air. Dia ada banyak air. It have many water at the xylem vessel. So this is the pulling process. You can see it is pulling process. It is not a pushing process. It is not say that because of there are many solute potential inside the mesosphere cell, so that water potential decrease, so that the water is pushed up. No, it is the pulling process because of the transpiration process. Okay, so the xylem, finally it will reach the xylem. So the water from the xylem is pulled into the surrounding cell and air space to replace the water that was lost. So first step is air space first. Because air space containing many water vapor, so it already lost out through the atmosphere, so that the water form that cope with the mesophyll cell starting to lose as well, evaporate as well, so that there is more doing more curvature and increase the surface tension. And due to this surface tension, it will create the pulling force to pull the water either from the fans for the surrounding mesophyll cell or the airspace to replace the water film. And finally, it will pull the water from the xylem vessel in the leaf so that it can replace all the water loss. And this pulling process, pulling process will reach to the stem of the plant and evenly until root of the plant. So this loss of the water will okay. Loss of the water will cause the xylem vessel pull the water from the root to the top of the plant. On the plant. Why? Because the water in the xylem also decrease. Because we want to replace, meaning that the xylem also do not have water anymore. So the water potential starting to decrease already. The negative water potential. So the water will xylem in the xylem is pulled into the surrounding cell and airspace to replace a water loss. So negative potential could provide the transpiration pool. So this is a key. So because of the water potential in xylem is already decreased, so it will create the transpiration pool to the leaf, in the leaf. Okay, so the transpiration pool is transmitted along the way of the xylem vessel that can be extended down until the roots. And the key component of the cohesion tension theory is that the water molecule is transported in unbroken change. It is pulling in a change one. There is not that the water is transported independently. No, it is unbroken. Meaning that why it is unbroken? Because of adhesion force and cohesion force that you still learn in chapter 1, water. What is adhesion force? Adhesion force and cohesion force. Because cohesion force is force between the water molecule, and adhesion force is force between the xylem wall and water molecule. And due to this cohesion and adhesion force, adhesion force make the water is transported without unbroken change to be pulled up narrow region and trachea or stem xylem. This in turn, the pull water up the root xylem, forming a continuous column of water from root xylem to the stem xylem and finally to the leaf xylem. So this is the overall process that about the cohesion tension pooling and also the transpiration pool. So the tension will cause the water potential in the root xylem decrease so that more water can flow positively from the soil into the root xylem with the aid of the cohesion and adhesion force. So the most important thing is at here. Cohesion and adhesion force is very normal. You need to know how the transpiration pool process is generated. Because all the initial process, if you want to generate the pulling force, is because of this. Because of this pulling force, so that it can pull the water from the root to the leaf of the xylem without any broken chain. It pull up continuously. And why this continuously occur? Because of the adhesion force and cohesion force of the water. So this is a key point that you need to know. Okay, so this is a summary up. So you need to see that when the water loss is water loss by through the somata, the water potential of the mesophyll cell decrease, hydrostatic pressure decrease, and negative pressure, surface tension is occur so that it generate the pulling force. So when the pulling force, so it will pull out the water from the root xylem to the leaf xylem. So when the root xylem water is decreased, so that the water from the soil will diffuse into the root xylem with the aid of the cohesion and adhesion force. So all this process. So the most key important thing is the continuous process. Cohesion tension theory is the water is transported continuously without any broken change. And then, okay, this is about the importance of transpiration. So why transpiration is important? So, of course, it is one of the transport process. It provide the water and mineral to the plants, and provide a cooling effect, play a major role in the water cycle, and through the decrease the temperature, and then for the photosynthesis, gases exchange, all that. Yeah. Okay, next is about the translocation, nutrient transport in plants. Okay, so still same, you need to know about the phloem cell, phloem tissue, sieve tube, companion cell, fiber, and parenchyma. Okay, so translocation, so these are terms that used in the transport of the product of the photosynthesis from sugar source to sugar sink by phloem tissues. Okay, phloem tissues. Okay, so you need to know what is sugar, what is sugar sink, what is sugar source. So sugar source normally is the place that is the product of the photosynthesis. So you need to know where is the photosynthesis occur. So normally it's occur at the leaf, right? So sugar source normally is leaf. And sugar sink is the place that require nutrient. So normally it's like the root, normally it's root lah. Root part of the plant. Okay. Okay, so in STPM, we will learn about the pressure flow hypothesis in translocation process. Okay, so first of all, so this is the sugar source and sink lah. So source is the process, is the terms that we use that region in the plant where the photosynthesis occur lah. Photosynthesis occur and organic solute asynthesis. So normally it's green leaf lah. So sink will be many area lah. It is an area where this nutrient can be loaded out, unloaded from the sieve tube. So normally it's root, foods, all that lah. So sink can be have many part, many regions. So in examination, if the question have already mentioned about the specific region, you need to mention the specific region. You cannot write a general term. If you write the general term, there will no marks. Okay. Okay, so sucrose load into the sink from source. Okay, so how the sucrose is transported from the leaf, from the leaf part into the xylem vessel. We know that in leaf we have many cells, right? And this other cell can do photosynthesis. And then after that, this all the produce organic nutrient, glucose molecule, all that will be transported into the phloem tissue, which is a sieve tube lah. So how this process occur? Okay, so similarly to the water transport, there are also two pathway that similar like the water. The first one is the Simplus and the second one is Apoplus. No, there are no vacuolar because vacuolar is majority for water only. So because this is not transport water, so there are no vacuolar pathway. So there are only Apoplus and Simplus. So Simplus is passed through the cytoplasm, through the plasmodesmata, similar to water, and Apoplus is through the cell wall. Through the cell wall only. So normally through the cell wall only. Okay, but after that, Apoplus, as I mentioned before, Apoplus. Okay, but in differences between the water and the nutrient part, normally your sucrose is transported through the Simplus pathway, and at the some of the region, when it is near the sieve tube, as you see near the sieve tube, some of the nutrient was starting to transfer through the Apoplus pathway. Some only. So Apoplus pathway is not transport from along the mesophyll cell to the sieve tube element. No, at the initial part is the using the Simplus. After that, when it near the sieve tube element, it was starting to use some of the nutrient was starting to use the Apoplus. Okay, Apoplus. Okay, so when you go through the Apoplus pathway, Okay, the sucrose that travel in apophis will be accumulated nearby the sieve tube or directly through the campaign cells. And in most part, the sugar movement into food require active transport. Active transport. So it is the core transport process if you want to transport the sucrose from the campaign cell into the sieve tube element lah, especially for apophis pathway. No, past 5. So what it call transport process, actually, you already learned in Sem 1 chapter 4 lah. So I think I don't need to explain more detail about the core transport. Okay, so next is about the pressure flow hypothesis. Okay, once the sucrose, so normally the nutrient is sucrose using the example. Once the sucrose is already enter the phloem sieve tube of the leaf, it was starting to transported through translocation process, which is a pressure flow hypothesis. Okay, so what is this happening? Okay, when the sucrose is already entering to the actively loaded into the sieve tube element that require ATP, which is a core transport process. Okay, it is already all the sucrose will be loaded at here, accumulate at here. Okay, so you need to know sucrose is highly when the sucrose is highly concentrated, is the solute. Right, it is solute. It is not a liquid like a water one, can flow very smooth one. So sucrose require a medium that can be transported along the sieve tube element. So one of the medium is water. Water is the medium that can be transport or anything, right? So it require water. So where is the water come from? Water will come from. So the water come from the xylem vessel here. We know that xylem and phloem normally is located side by side only, right? So water is required from the xylem vessel in sieve tube. So at here, when you accumulate the solute sucrose at here, at this region, meaning that water potential starting to decrease. So the water potential in the sieve tube will is increased, so the water will be diffused out from the xylem vessel and entering into the sieve tube by osmosis process. Okay, osmosis process, so that the hydrostatic pressure now increase, so it able to transport the sucrose from the source region into the sink region. So the increased pressure force the fluid through the sieve tube to the root, to the fluid, to the shoot, or to the stem, to the fluid area, all that also can. Okay, after it reach the sink region, for example, is blood lah. Okay, when it enter the it reach the sieve tube that near the sink region, the sucrose now is actively transported into the sink cell, which is the process we call as unloading. Unloading process. So it require ATP as well. ATP. Okay, so when the sucrose is already actively transported into the sink cell, meaning that your water potential at here is increasing. Because water is not transported into the sink cell one. Water is still only maintained into the sieve tube element just for the transport medium to transport sucrose from the source to the sink only. So when the water increase, so this water will diffuse back from the phloem sieve tube element to the xylem vessel. So once water is in the xylem, transpiration pull the water to the leaf again, and this recycling process happen again and again. So you can see here. So it is very simple. When the sugar is accumulated here, it require water. Water come from where? From xylem vessel. So water enter into the sieve tube and it transport to the sink region. After that, this sucrose actively unload the sucrose to the sink cells, and the water increase, so the water recycle back to the xylem vessel, and the xylem vessel will pull the water up again, and once at the source, sucrose still will, when there are the sucrose concentration high, then the water will come back, diffuse back again. So this recycling process. So this is what we call as pressure flow hypothesis, based on the pressure, based on the hydrostatic pressure of the sieve tube element, so that it can transport the sucrose from the source region into the sink region. So, next is about the experimental setup between the shown in the diagram below. So they are about the pressure flow hypothesis. So the previous one is about the real situation, real phenomena, and this one is the experimental setup by one of the scientists. I already forget their name, his name. So actually his setup is about the mass flow hypothesis lah. Pressure flow hypothesis is about the real situation, but this setup actually is called as mass flow hypothesis. But both of them are acceptable when you want to use it in examination lah. Okay, so for this diagram, normally it come out in objective question. Okay, you need to know what is this, what is this, what is this, what is this, in based on the plan. So A and B, either it is sink or source. You need to know either it is the sink or source. So sink characteristic is that it produce not produce, it receive the sucrose. It want the sucrose one. And the source is the place that produce sucrose. So you will see this diagram easily because it's using the color. In real examination, normally either it got label or it not label, it just label the water direction only. Just label water direction, and then you need to know, or they already label here A and B, but no water direction. You need to predict the water direction. Yeah, something like that. Okay, so for here, you clearly mentioned that A is the source, B is the sink. So what is this tube? And what is the water surrounding? So normally, this tube is the sieve tube. This is a sieve tube. We consider as sieve tube. And this is the region of xylem vessel. Xylem vessel. The water here is xylem vessel. So, okay. When we see to the A, when they are A, so we know that A is source. So source is highly concentrated. The solute potential is very high. Their sucrose concentration is very high. So meaning that water potential is very low. So the water from the surrounding area here, like xylem vessel, here will diffuse into the A. So A is the place we call as the sieve tube element near the source. Source. Okay. So water potential is diffused into it. So when the water potential increase, so that it will transport the sucrose from upstairs and then into the B. Okay. At the same time, B, because is less concentrated, water potential is very high. It's very high, right? It's very high. So water is diffused out from the B. So when the is diffused out from the B, forced by a hydrostatic pressure. Okay, so when the when the sucrose is entering into the B, because this is the experimental setup, so the sucrose will remain at there. Sucrose will not diffuse out because in real situation, in pressure flow hypothesis, real situation, sucrose need to actively transported, unloading from the B. But because of this experimental setup, it is normally based on the down the concentration gradient, so the sucrose will not be transported but the water will be transported out. Because at the beginning of the part experiment, the water potential is now here is still very high. Because all the water is already go into the A. So the water potential at the surrounding now starting to decrease. But when the sucrose starting to accumulate at the B, of course the water direction for the B is very less and finally this one will reach the equilibrium. Yeah. But mass flow hypothesis, this experiment just want you for you to know how the sucrose is transported from here based on this theory. Yeah. So that's all about this chapter and also this video. So Thanks for those watching from the beginning until end, and thanks for your supporting. Hopefully you guys can score well in STPM Biology, especially in this chapter. So I already try my best to provide the free class learning to you all. So hopefully you guys can get it what I am teaching about this. If you have any question, you just comment about it or join the Telegram group as well. So that's all about this video. Thank you and bye.